I'm Trudi Schupbach. I'm a Howard Hughes investigator and a professor of molecular biology at Princeton University. In my laboratory, we are interested in the question how pattern and polarity is established during development. One of the big problems in developmental biology is the question of how we go from an egg that contains a single fertilized nucleus to an adult organism that is complex and consists of many different cell types. The model organism in which we study this question is Drosophila, the fruit fly. Here in the upper panel you see a fertilized egg just as it's been deposited by the female, and it has a single nucleus inside. Later in development, as we go through development, we end up with an adult fly which has a head, a tail, has wings and legs. And the question then is, how do we go from the egg with a single nucleus to this complex organism where different cell types have nuclei which now express different sets of genes? It has become clear over the past 20 years or so that eggs are actually not just simply unstructured cells, but that there is a lot of organization within eggs as they are being produced by females. And in particular, there are some important informative molecules that are often localized within eggs. So for instance, here on the upper panel is again an oocyte of Drosophila, and you can see a key molecule, the Oscar RNA, which is accumulating at the posterior pole of the egg. On the lower panel, you see an amphibian oocyte. And in this oocyte, you can see a key RNA molecule, VEG1, localized to the vegetal pole of the oocyte. If we want to understand how these eggs are built in these structured manners where molecules are localized to one end or the other, we have to look back in oogenesis. This picture here shows you an ovary of a fruit fly dissected out of the female, and you can see that there are many egg chambers here that are developing. At the top we have the youngest egg chambers, and as they develop they move to the posterior, so that we have the oldest egg chambers down here. If we just look now at a single one of these strings of egg chambers, an ovariole, we can look at this in more detail. At the top of the ovariole we have stem cells. These stem cells divide continuously and they give off daughter cells which will differentiate into the eventual eggs. Now in insects there is a little step in between the stem cell division and the actual differentiation of the egg. The daughters of the stem cells will undergo 4 more mitosis, and they will form a cluster of 16 cells. 15 of these cells will become nurse cells, and 1 of the cells will become the oocyte. This cluster of 16 cells is then enveloped by the follicle cells, and the whole entity is called an egg chamber. And it's within this egg chamber that the pattern and polarity are established. So in Drosophila, both the anterior-posterior and the dorsal-ventral axis can already be seen in the egg after it's been laid by the female. So we know that, for instance, the head of the embryo will form in this part of the egg, and the tail here, and then the dorsal and ventral side can also already be predicted when we look at this egg. So the asymmetries are already visible in the egg chambers during later stages of oogenesis. So here in this picture here, you see an egg chamber, and you can predict the anterior-posterior and dorsal-ventral axis because the oocyte is always at the posterior end of the egg chamber, and the oocyte nucleus occupies a dorsal position within the oocyte. And whichever side of the oocyte nucleus is closest to, that will be the future dorsal side of the egg and the embryo. Now these asymmetries that are visible in the egg chamber are then also seen in the egg shell. this is what we are really looking at here, the outside of the egg shell. And the egg shell is formed during oogenesis by the follicle cells which surround the oocyte. What this means is that the follicle cells have to have also a pattern information because they have to form this asymmetric eggshell in correspondence with the asymmetries that are present in the oocyte and nurse cells. Now, how can we find out the underlying molecular processes that establish these asymmetries, and how can we understand how the follicle cells know where the anterior and posterior end of the egg are going to be? In using a model organism, we have the advantage that we can, when we have such questions, that we can do mutagenesis experiments and let the organism tell us what genes are important for these processes. What we can do in Drosophila is to perform a mutagenesis experiment by feeding males a mutagen. We can then cross these males to females, breed such lines through some generations until we get females that are homozygous for a particular mutagenized chromosome. And then we can test such females to see whether they lay normal eggs, whether they lay abnormal eggs, what the embryos look that are inside these eggs. And whenever we see something of interest to our question, we can retain the lines and then study them in more detail. In this slide here, I'm just showing you a panel of different mutant phenotypes that we found when we did such mutagenesis screens. On the top here is a wild-type egg, again, for your reference. You remember it has a dorsal side, and the dorsal side of the wild-type egg is particularly characterized by these two structures, the dorsal appendages, that protrude from the egg surface. They serve as a good landmark for dorsal-ventral pattern formation. So here in the middle, for instance, then you see an egg that doesn't have any dorsal appendages. It's a completely ventralized egg. It has lost all its dorsal structures, and when this egg is fertilized, the embryo inside will also be ventralized and will only form ventral structures such as mesoderm and ventral ectoderm. Conversely, here at the bottom is an egg that is dorsalized. Here, instead of just having very defined groups of follicle cells on the dorsal side that form the dorsal appendages, here on the bottom, follicle cells from all around the egg circumference have become dorsal and have migrated and have tried to make dorsal appendages. When these eggs are fertilized, the embryos inside are also dorsalized, and they form only dorsal skin and no mesoderm and no ventral ectoderm. However, these phenotypes pose a problem because we see a change in patterning, in fact, here, both in the egg shell, which is formed by the follicle cells, and by the embryo that forms inside the egg itself. And whenever in development we have mutations that affect two different tissues, we have to ask, well, then where is the wild-type gene really active in development? Is it in the follicle cells? Is it in the egg itself? Or maybe in both tissues? Where is the pattern established first? What comes first? What comes second? And there are methods to ask these questions that we use a lot in development, and they usually involve making mosaics. In this particular case, where we're interested in the question of patterning between the egg itself and the follicle cells, we can make mosaics where one is mutant and the other wild type. Here, in this example, we're using transplantation of cells. We can transplant in Drosophila germ cells at an early stage from one embryo to another. So we can, for instance, take the germ cells out of a mutant donor embryo and implant them into a wild-type recipient embryo. Then this wild-type recipient embryo grows up into a fly, it will be itself, in most of its body parts, wild type, meaning normal, but the implanted germ cells will then give rise to mutant oocytes. And so then we end up with a mosaic where we have a mutant oocyte and nerve cell surrounded by wild type follicle cells, and we can then ask what will happen. We're going to look now at the results that were obtained by such pole cell transplantations for two of the mutants here that ventralize the egg and the embryo. They're called Torpedo and Gurken because of their mutant phenotypes. Performing such transplantations, we found in the case of Gurken that when we had a mutant germline but surrounded by wild-type follicle cells, we saw the mutant phenotype. The eggs and the embryos were ventralized. Conversely, when we transplanted wild-type germ cells into mutant female hosts, we found that in this particular case, then wild-type eggs and embryos resulted. This leads to the conclusion that the Gurken gene is required in the germline. Whenever the germline is mutant, mutant eggs and embryos result. Whenever the follicle cells are mutants, that doesn't really matter. Now, very interestingly, when we did the converse or the same experiment with the other gene that also produced ventralized eggs and embryos, Torpedo, we found the opposite result. Here, it appears that Torpedo is required in the follicle cells because in the transplants, we only see the phenotype, the ventralized phenotype, when the follicle cells are mutant. We don't see it when the germline is mutant. This suggested that there would be some sort of communication going on between the germline and the follicle cells, because Gurken is obviously doing something in the wild-type germline that is important for pattern formation, but it's not only important for the embryo that forms inside the germline, it's also important for the surrounding follicle cells. With respect to Gurken, the follicle cells don't seem to look at their own genotype for pattern formation, but whenever the germline is mutant, they will form an abnormal eggshell, even though they themselves have a normal genotype. That then suggests that whatever Gurken does in the germline is somehow signaled to the overlying follicle cells. And Torpedo, which is required in the follicle cells, might then be acting in the follicle cells to receive this information that comes from the germline and help the follicle cells to act and implement this information. These results were further corroborated after we cloned the genes and found out what the proteins are that are encoded by Gurken and Torpedo. Torpedo, we found, encodes the Drosophila homologue of the epidermal growth factor receptor. So this is a receptor molecule that's very large. It sits in the membrane of cells and it can bind ligand molecules. In fact, the EGF receptor is required multiple times during Drosophila development, and it acts in several different tissues. It can be activated by a number of different ligands. Gurken, which is one of these ligands, is specifically used, though, by the oocyte to signal to the EGF receptor during oogenesis. Very excitingly, when we looked at the protein distribution of these two genes, we found that the Gurken protein accumulates very asymmetrically within the oocyte. So here on the upper panel, you see the expression pattern of the EGF receptor, which is expressed in the follicle cells. It's expressed in all follicle cells, dorsal follicle cells, as well as ventral follicle cells. Gurken, on the other hand, you can see accumulates here in a very restricted part of the egg chamber. It's just found at this stage on the dorsal anterior, in the dorsal anterior corner of the oocyte, close to the oocyte nucleus. Remember, Gurken encodes a signaling molecule, and this can then bind to the EGF receptor. So now we can understand that the oocyte, which has localized its oocyte nucleus to one side of the egg chamber, uses Gurken now to signal the position of the oocyte nucleus to the overlying follicle cells. These follicle cells that see high levels of this Gurken molecule will activate the EGF receptor, and that will lead to the expression of certain target genes. These target genes will not be activated on the ventral side, where the follicle cells don't see high levels of the Gurken protein. So we can start to understand how this dorsal-ventral asymmetry that arises in the germline is then signaled to the follicle cells and is used to create dorsal-ventral asymmetries. Of course, we then have the question, well, if we see the Gurken protein localized, how is it? Why is it just found in this particular part of the oocyte? Well, the Gurken protein is at the anterior dorsal corner at this stage because the Gurken RNA is localized to this corner. So the nerve cells transcribe the Gurken RNA, it's rapidly transported into the oocyte, and then it's accumulating in this corner close to the oocyte nucleus. After we did some more analysis of the mutant phenotypes and also of the protein and RNA distribution, we learned that in fact the Gurken EGF receptor signaling pathway is used twice in oogenesis. Not only at this later stage where Gurken indicates the dorsal position of the oocyte nucleus to the follicle cells, but even earlier in oogenesis, Gurken is already active. So in this uppermost panel, you see in the small oocyte, the Gurken protein already being expressed. The oocyte at this stage is very small, and Gurken signals at this stage to the follicle cells at the posterior end of the egg chamber. It induces those follicle cells at this stage to be posterior. The posterior follicle cells then signal back to the oocyte, and this signal is still not quite well understood, but we know that the signal back to the oocyte from the posterior follicle cells results in a reorganization of the microtubule network that causes the oocyte nucleus to move off of its axis of symmetry and over to just one side of the egg chamber. Once it's in its asymmetric position, the Gurken RNA and the Gurken protein then follow the oocyte nucleus, and now Gurken signals again through the EGF receptor to the follicle cells, but this time the signal indicates to the follicle cells to become dorsal rather than ventral. The fact that Gurken signals early to establish the anterior-posterior asymmetry within the egg chamber is illustrated here in these in situ hybridization pictures, where we look at particular anteriorly or posteriorly localized molecules. So one very famous anteriorly localized RNA in oogenesis is the bicoid RNA, which you can see here in the wild type just accumulating at the anterior end of the oocyte. But in mutations in which Gurken EGF receptor signaling is blocked, you can see down here that now the bicoid RNA is found both at the anterior and the posterior end. Conversely, the Oscar RNA, which is normally just localized to the posterior end of the egg chamber and egg, in the mutants, doesn't seem to know where the posterior end is and instead accumulates somewhere in the middle of the oocyte. So then that tells us that we need the Gurken EGF receptor signaling pathway early on to establish the right anterior and posterior addresses so that these RNAs know where to go. To summarize this, we can look at this cartoon. So early in oogenesis, we have a first signal where Gurken signals to the EGF receptor and induces the posterior follicle cells to be posterior. The posterior follicle cells then send the signal back to the germline. This signal, as I said, is at the moment has not been totally determined in molecular terms, and we in our lab and other labs are trying to find exactly how this happens. We do know, though, that this posterior signal then results in the reorganization of the microtubule network within the oocyte, and also causes the oocyte nucleus to move to one side of the egg chamber. And then once the nucleus has moved, we have the next stage of signaling where Gurken again signals through the EGF receptor to the dorsal follicle cells and induces thus the dorsal-ventral pattern of the egg and embryo. In summary then, I've tried to explain to you in my talk how in Drosophila both the anterior-posterior and the dorso-ventral asymmetry of egg and embryo are established during oogenesis. The germline initially makes these asymmetries by moving the oocyte to the posterior and moving the oocyte nucleus to one side, but in both cases these initial asymmetries are then signaled to the follicle cells through the Gurken EGF receptor signaling pathway. The follicle cells are induced to take on specific fates, and the follicle cells then signal back to the oocyte and cause asymmetries within the oocyte. And so by signaling back and forth between the germline and the follicle cells over the time of oogenesis, both major body axes of the egg and embryo are established.